So we'll go ahead and get started. So just a reminder, this is for Bio 1 with Dr. Thomas. This review session will be from 1 to 3 o'clock and will also be uploaded as a video later for you guys to see as well. All right. All right. So if everyone's ready, we'll go ahead and get started. So I have uploaded this document. It's up on both the Study Union WordPress site and the UCF-SI WordPress site. I will also be uploading the answers to my WordPress site after the review, in case you missed anything. All right, so if everyone's ready, we'll go ahead and get started. Your exam with Dr. Thomas, she said should be about 60 questions. I've made a review for about 80 questions to kind of give or take some possible areas you guys might need some extra help. So the first section that you guys had for the course was talking about some general chemistry ideas. So we'll go ahead and get started with that section. So for question 1, which of the following statements concerning protons is true? Just kind of a basis question. So for A, we have protons are molecules found throughout the atom. How do we feel about that? Is that true? A little response would be nice too. Okay, thank you. So that's not true. How about protons are small neutral particles? Good. So protons are not neutral. They should be positive. All right. How about protons are found in the nucleus of an atom? All right. So that one is true. And how about D? Proton number cannot be used to identify an element. So that is also not true because proton number can be used to identify an element. All right. So for number 2, an atom of an oxygen has 10 neutrons. What would this be known as? So in the case of periodic table, if we were to be able to see that, so oxygen is number 8. That means that oxygen should have 8 protons, 8 neutrons, 8 electrons. So in the case, since we have 10 neutrons in this case, should it be considered a different element, an isotope, a molecule, mutation, or an ion? What do you think? Good. So I heard isotope, and that'd be correct. All right. All right. Number 3, a blank is an atom that has gained an electron to fill a valence shell. So in the case, if we're gaining an electron, should we become more or less negative? So we're becoming more negative. And in that case, we should think of that as an anion or a negative ion, whereas our answer E would be if it were to be positive. All right. All right. So for number 4, we're getting a little more into structure and bonds. So which of the following is true? Covalent bonds transfer electrons while ionic bonds share. Is that true? No. How about, do both of them share electrons? Good. So I see some heads shaking no. Both covalent and ionic bonds have polarity. It's not always true either. So D, covalent bonds share electrons while ionic bonds transfer. How about that one? Good. So this one is true. All right. So number 5, really important. We've talked a bit about our hydrogen bonds. So hydrogen bonds, they can be found in several places. Most particularly, we're going to see them in between DNA strands. All right. And in the case for where are they always found? So through our answers, should they be found between 2 partially charged molecules? What do we think? Okay, I see some maybes, so let's look at the other ones. How about, is it between 2 oxygen atoms? So just a little chemistry tip. So hydrogen bonds should be hydrogen with nitrogen, oxygen, or fluorine. All right, so hydrogen bonds, we at least have to have the hydrogen aspect. And so in the case, this is really going to be looking for where we have a difference in polarity. Think again, so water molecules. So in the case of our water molecules, it won't be between two oxygen atoms. It won't be between two positive ions. It won't necessarily be between any compound containing hydrogen. It has to have nitrogen, oxygen, and fluorine. And then in the case of water and a nonpolar molecule, should we see a polar molecule mixed with a nonpolar molecule? I heard someone. So no. In the case, if it was a polar molecule, water can go ahead and bond. All right. So in the case, our only one that's left is A. All right. And so just really quick, I wanted to show you guys just a reminder sketch. So in the case, if we were to kind of draw out water, if are looking at the central one, it will go ahead and bond, so our negative to positive. And in the case of a water molecule, we should have 4 hydrogen bonds occurring for one water molecule. All right. All right. So in the case, we also have to define organic chemistry. So organic chemistry is basically what? The chemistry of what, guys? Good. So it's our carbon chemistry. So C, chemistry of compounds that contain carbon. All right. So also kind of review what makes up 96% of the elements living on Earth, or not living on Earth, of living things. So we had 2 ways we could think about it. We had honk, or we could think of it as chan. So this would be our carbon, hydrogen, oxygen, nitrogen. So in that case, that would be answer B. All right. Yes. The other 4%, that's a great question. So our other 4% was, does anyone happen to remember off the top of their head? No? All right, so our other 4%, we had potassium, we had calcium, we had sulfur. All right, starting to sound familiar. So this can, and potassium. All right, so this can also be our P cask. All right, so that would be our other 4%. All right. All right, so in the case, if we're focusing on carbon, so again, kind of thinking back to our organic chemistry, what type of bonds should carbon form? So in the case for carbon, should we have covalent bonds? So I see some head shaking yes. So carbon's always going to at least form a covalent bond. Is it going to form hydrogen bonds? So again, this would be no, because it has to be hydrogen with nitrogen, oxygen, and fluorine. So polar bonds is just another type of covalent bonds, but that's not necessarily what carbon's always going to form. And then, so this would not be always. And in the last case, ionic bonds would just be even more extreme versions of polar bonds, and carbon is not likely to form ionic bonds. All right, so further structures for amino acids. Amino acids are important in the formation of what? So Are amino acids important for nucleic acids? No. How about codons? Good. How about proteins? Okay, good. So I see some yeses. How about carbohydrates? Are they related to those either? Okay, good. So just kind of also point out amino acids can kind of be interrelated. So remember that we do have nucleic acids, right? Where do we find nucleic acids, guys? So these help create our nucleotides. All right. How about nucleotides? Where do we find those? Thank you. So I got some DNA and RNA. All right. And then, so if we have 3 nucleotides, what do we call that? So if we times that by 3, this can help us make our codons. All right. And codons help us do what? So when we go through our codons, these are going to help us create our amino acids. And lastly, our amino acids, when we put them all together, what do they make? Good. So I heard proteins. Okay? All right, so that's the structure. So some of these things may be interrelated. However, amino acids, they're in the step next to creating our proteins. Okay? All right. So what is the general structure for amino acids? What do we have inside? So we at least know we're going to have a central carbon since it's in all of our answers. How about the final groups? So this is kind of remembering what kind of functional groups that we had. So in particular, for an amino acid, we should also at least see an amine. However, the last part is where we're going to have to make our differentiation. And in the case, so we should also have a carboxyl group. All right. So just to kind of remember what those look like, our amine is NH2, and our carboxyl is COOH. Okay. All right, so some different aspects behind chemistry. We also have our pH, so definitely be able to understand the few different ways we can manipulate pH. So in the case of a pH being 7, what should the hydrogen ion concentration be? So should we have 10 to the positive 7? Should we have 7 times 10 to the 1? How about 10 to the negative 7? Good, so 10 to the negative 7, all right? This could also be 1 times 10 to the negative 7. So if you see in either format, both would be correct. Right? All right, so in the case for number 12, so we have an OH ion concentration. So in this case, we have 3, 6, 8. All right, so we should also be able to say that this is 1 times 10 to the negative 8 for our OH concentration. And so in that case, our POH should be what? Good, so our POH in that case should be 8. And does that mean we are in an acidic environment or a basic environment? Good, so this should be an acidic environment. So also remember that in this case, so if we have our spectrum, all right, so if we have 7 in the middle, we have acid over here, we have a base over here. Our pH should be 1 to 14, and our POH should be the opposite of 14 to 1. All right. All right, so then in the case for number 13, all right, we see that the OH concentration, all right, here, and this is the same as 1 times 10 to the negative 5. All right, so our POH in this case should be 5. And then does anyone remember what the equation is to get pH from POH? All right, so in the case, our pH and our POH both together should equal 14. So here, our pH should be 14 minus 5. All right, and so overall, our answer should be 9, and we should have a pH of 9. All right. All right, so next we have number 14. So what are the 4 major classes of macromolecules in living systems? So what do we think? What do we need? All right, so we will need our carbs. These are our sugars. All right. We'll need lipids. All right, these are our fats. We will need our proteins. And we will need to be able to make these proteins, so we'll also need our nucleic acids, all right, which will also help form other secondary structures. All right, so in that case, that is most related to answer C. So again, be careful you know where nucleic acids versus amino acids are located, how they're created. All right, don't get tripped up between those two. All right, and we also need to remember some of the details about structures. All right, so when we have 2 or more finished proteins associate together, what type of structure should we be in? So this will be our final structure or our quaternary structure. If we see the presence of alpha helixes and beta sheets, what should we have? So this will be our secondary structure. All right, a sequence of amino acids. Good, so this will be our primary. And then the final folding of the functional protein should take place where? Good, so I heard some tertiaries. All right, so just a few kind of important notes here if you have a hard time kind of thinking about some of these things. So if we're thinking about primary, we can think about our DNA being as a structure, or in the case, our DNA being single-stranded. All right, so that also means our RNA can be a primary structure. If we're looking at alpha helix, so our DNA double helix is similar to our alpha helix. And then if we want a quaternary structure, we'd have to go larger molecules. In the case, most commonly, we'll think of hemoglobin. Okay? That has several proteins associated together. Tertiary is generally just a final protein. Usually not too many examples are given for tertiary because we usually see them as associated to one another. Okay? In the case for which of these are held together by hydrogen bonds, what should we have? So if we think about some of our examples, where do we see hydrogen bonds helping us hold together? Good, so our secondary. All right, so in between our DNA base pairs, all right, when we start to make the helix aspect for DNA, that'll be our hydrogen bonding and our secondary structure. And then, so which of the following are held together by peptide bonds? Good, so this will be our primary. And which are held together by R groups? Good, so tertiary. Okay. So all functional proteins have which of the following structures? So do they all have primary structure? Good. How about secondary structure? Good, so we should also have secondary. How about tertiary structure? Good. How about quaternary? Do we need it to function? Good, so we don't need quaternary structure. All right. So next in the case, we had our chaperonins. So our chaperonins were guys that kind of looked like this, and they had a cap on top. All right, so this cap would open up, it let something inside. In the case, what is the function of a chaperonin? Is it condensation of monomers to polymers? No. How about hydrolysis of polymers to monomers? How about assisting folding of polypeptides? Good. So also remember, polypeptides, the same thing as our proteins. All right. Does it also work in anything else? Is it just proteins, or do we also need it for, say, lipids? No. All right, it's just going to be our proteins. Our proteins are the only things that are going to have to fold. All right. And it's also not going to help with transport. It's just going to stay where it is. All right, so we also had our section about organelles and structures. So it's important to kind of remember what these structures are and where they might be located. So our main ones we have here is our nucleus, our nucleolus, our lysosome, and our ribosomes. So what is the control center for the cell? Good, so our nucleus. What synthesizes proteins? Good. So that will be our ribosomes. What participates in intracellular digestion? Good. Our lysosomes. Which of the following can partner with endoplasmic reticulum? Good. So that will be our ribosomes. And in the case, all right, this will help create our rough ER. And then, so which of the following is located inside the nucleus? So, good, our nucleolus is located inside. All right. What else is located inside the nucleus? Good, so our DNA is also located inside the nucleus. It's going to be basically the house. All right. And then, which helps assemble ribosomes? All right, so this one's a little more not commonly talked about. So the assembly for our ribosomes is going to be our nucleolus. All right. So in that case, we can also assume our ribosomes are created inside the nucleus. All right. So from there, they're either going to attach from the rough ER or to the rough ER or be free ribosomes. We also had some questions about our cytoskeleton. All right. So in the case, which of the following is incorrect concerning the cytoskeleton? So both our microfilaments and microtubules help maintain cell shape. Is that true? So that is true. All right. cytoskeleton, kind of think about your skeleton. It helps you maintain your shape. Both microfilaments and microtubules assist in intracellular transport. What do we think? Good, so that will also be true. Both microfilaments and microtubules have functions in the cytoplasm. Good, true. We normally find them in the cytoplasm, so we hope to expect them to function there. Both microfilaments and microtubules assist with chromosome movement. What do we think? So that one is not true. In this case, only one of them is going to assist in chromosome movement. Which one do we think? All right, so microtubules will help us with our chromosome movement. All right. And then, so do both microfilaments and microtubules, are they similar in size? What do we think? So that one's also not true. All right. So they're on the level that they are size-wise, they're considered to be fairly different. So in the case, our microfilaments are 7 nanometers and they form dimers. All right. And then our microtubules are 25 nanometers and they form a tube. All right. So there's also some other things that she could ask in this case about the cytoskeleton. So for the case, it might be good to also note that our microfilaments, all right, these are made of actin, and our microtubules, these are made of tubulin. All right. There is a third type of cytoskeleton. Does anyone remember what it's called? Good. So we also have our intermediate filaments, all right, and this guy is intermediate in terms of pretty much everything. He's going to be intermediate in size, going to be in between both of those types of things on structure, all right. This guy will also be in things like what we consider like our hair and nails, okay? All right, so which of the following incorrectly matches the structure with their function? So gap junctions use channels to form continuous cytoplasm between cells. Is that true? All right, so remember our gap junctions are our most commonly thought of type of junction in terms of proteins. So it's going to help form kind of like our protein channel between cells, and that will also allow us to have continuous cytoplasm. So this one is true. In the case of tight junctions, do they allow cells to share adjacent cell membranes? So that one's not true, all right? Tight junctions are usually found in like the lining of, say, your stomach, all right? So these guys, they don't share the cell membranes, but they make sure that their cell membranes are tightly posed next to one another so as to not let anything pass through them, okay? So think again, if like your stomach, if things could pass through, that'd be terrible for you. So desmosomes do they allow anchor cells to one another using filaments? What do we think? So I see some head shaking, yes. So this one is also true. So kind of think best ways to remember this is that gap junctions are for channels, desmosomes are for anchoring, and tight junctions are to keep everything close and tight. All right. All right, so next we have our transmembrane proteins can function as both passive and active transporters. Is that true or false? What do we think? Good. So this will be true. All right. So our cell membrane, remember our transmembrane proteins go all the way through. It doesn't really matter what type of transporter it is. It will be determined by what we need to have transported. All right. So that will be determined by a few other things, and we will talk about transport in just a little more in depth in just a second. So for passive transport, does it require energy? No. So that will be instead our active transport. Does passive transport include diffusion and facilitated transport? What do we think? So this one is true, all right? So diffusion and facilitated transport are both types of passive transport. And then, so does passive transport flow against the gradient or with the gradient? Good, so this one should be with the gradient if it's passive. All right. In the case, if it's against the gradient, what type of transport is that? Good, so that one would be active. And then, does passive transport produce ATP? So typically, we're going to say our passive transport does not produce ATP. All right. In the case, we do have one type of transporter that produces ATP. Does anyone remember what that transporter is called? So that transporter is our ATP synthase. All right. And in this case, this guy would be a passive transporter. All right. However, in the case, not all passive transport creates ATP. All right, just a very special one. All right. So in the case, real quick, what other types of membranes do we have that are associated with our plasma membrane? So we have our transmembranes. What else can we call those? So we can also see this referred to as integral proteins. All right. And then we do have one other type. However, this does not go through. It's simply on one side, or does not go completely through the membrane. And these are peripheral proteins. Okay? So just be aware through some of these questions, they may not involve all the different types. So if there's ever a point where you kind of know what some of the answer options are referring to, it would be a good idea to kind of go back through and kind of review that section, all right? So again, like both her list and some of the reviews, they may not be extensive in all the material. However, they do cover at least main points from the different topics. So if there's ever one that you don't know, make sure you go back and look through all the different materials. So in the case, if you didn't remember what a transmembrane protein was, you can go back and kind of think about the other different types so that you know which one has which function. Okay? All right. So which of the following is true about amphipathic structures? Do amphipathic structures have polar groups? What do we think? So they don't just have polar groups. Are they usually very large in structure? Good. So they're also not usually very large. We're usually going to see them on a cellular level. Do amphipathic proteins, do they contain hydrophilic and hydrophobic regions? Good. So C, they do contain hydrophilic and hydrophobic regions. Are amphipathic structures found in all life forms? So It's a no. We don't always find them in like certain prokaryotes or bacteria. All right, so remember those are particular types of life forms. And then, can amphipathic structures act as an acid? No. I like the thumbs up, thumbs down. It's really, thank you. All right, so amphipathic structures, again, amphipathic structures are very important for our transmembrane proteins. All right. Again, the structure allows the transmembrane proteins to sit within the plasma membrane like it does, all right, because our plasma membrane has the phospholipid bilayer, all right? So just remember that we may have, all right, so on the outside we're going to see our polar heads, where on the inside we're going to see the hydrophilic tails, the hydrophobic tails, all right? So in the case, we're going to want the transmembrane to have the ability to associate with either of them, all right? All right, so cholesterol inside the membrane primarily helps what? So it helps, So it can help us with stabilizing the cell, but that's not our primary function. So it's going to help us maintain fluidity, all right? It's going to help us have the semi-permeable aspects, all right? So without the cholesterol, certain things may not be able to pass through, say like certain ions, or sometimes, like in the case of water, certain things will not be able to simply diffuse through the membrane without it. All right. So one other thing that is particularly important, what type of molecules are efficient for diffusion through the plasma membrane? What do we think? Good. So A is small and hydrophobic. So these will be the most efficient because, remember, our diffusion, all right, usually occurs without proteins or without the help of proteins. All right. And in this case, it'll also be our passive transport. All right. So definitely remember where we might see some of these things occurring. She may throw a question in there asking you about different types of salinities. So just kind of remember we had a few extra vocabulary words to kind of referred to what type of solutions we had. So we had our hypotonic, our isotonic, and our hypertonic. All right, and remember, so when it's saying hypertonic, it's necessarily referring to what aspect of solution? So in the case, tonic is referring to kind of the salt aspect or whatever is being dissolved in the solution. So in the case of hypotonic, we would see low salt levels, all right, higher water levels. How about isotonic? What did isotonic refer to? Good, so isotonic was salt being equal to water. And in the case, our hypertonic is the opposite of the hypotonic. So here we'd have our high salts and our lower water concentration. And so in the case, what direction does water flow to? Good. So water is going to flow from higher to lower, all right? So in the case, if we have two types of solutions, and one of them is hypotonic and the other is hypertonic, the water is going to flow from the hypo to the hyper, all right, because it's going to want to come in and dilute the salts or go to the lower concentration of water, all right? So just kind of brief review. If you want some extra, remember she did have some really good notes about how to determine which solution was which, okay? All right. So we also talked a little bit about energy and reactions. So in the case, which of the following is correct about the first law of thermodynamics? So A, spontaneity must always increase. All right. So in that case, spontaneity, this is referring to one of our laws of entropy. All right. But not the law of thermodynamics. Does it mean delta G must always be negative? Good. So, all right, delta G doesn't necessarily always follow thermodynamics either. Energy cannot be created or destroyed. Good. So that is the law of for the first law of thermodynamics. And then all reactions in nature should be exergonic. What do we think? No. So that one's also not true. Just like delta G, it can be exergonic or endergonic. All right, there's no law saying which way it should go. And then anything in motion will remain in motion unless acted upon by another force. So that does not refer to our thermodynamics either, all right? All right, so kind of review a little more about the reaction types. So from these following descriptions, which properly describe the following diagram, all right? So in the case here, we are looking at our reactants, all right? So our reactants are over here. These are our products, all right? And so this is looking at, on this side, we're referring to our energy, all right? So in the case, is this an exergonic reaction or an endergonic reaction? What do we think? So in this case, we are putting out energy. So we have an exergonic reaction. All right, so here you're going to lose. This one will be absorbed if it were to be endergonic. All right, so the reaction in the case, so we are releasing energy. So, which also describes the relationship between the reactants and the products. So, do the products of this reaction have a greater energy than the reactants? No. How about the reactants of this reaction? Do they have a greater energy than the products? Yes. So we also talked a bit about enzymes. So which of the following is false regarding enzymes? All right. So are enzymes nonspecific? What do we think? So enzymes are specific. All right. So one enzyme to one reaction. All right. Can enzymes change the delta G of reaction? Good. So they cannot change the delta G. Can they lower the energy of activation for the reaction? Good. So this one is true. So can enzymes be depleted when we run a reaction? Oh, I hear some yeses and I hear some no's. So interesting, they cannot be depleted. So this is also false. All right. So if we put an enzyme in, the same amount is expected to come back out after the reaction. All right. Similar to if, like, think about the binding aspect, our enzyme is going to bind to only one type of receptor. And so in that case, the receptor is the part that's actually being used in the reaction. All right, so our enzyme's not actually going to be used up. All right, and then do enzymes slow down a reaction? Good. They should speed up the reaction. All right, so what are all enzymes made out of? So are they made out of polysaccharides? So just kind of a reminder, what are polysaccharides? So these are sugars, all right? And so enzymes never made out of sugar, all right? Are enzymes made out of lipids? Good, so they're not made out of fats either. Are they catalysts? Good. So they are always going to be catalysts. Are they always made of polypeptides? So this is, again, so this is our protein. So in the case, if we say that all enzymes are made out of polypeptides, this is actually false. In the last section, we learned about an enzyme that is not made out of a polypeptide. All right, so not always, but usually. All right, most of them will be polypeptides, but not all of them will be polypeptides. All right. So, and then are any enzymes made out of nucleic acids? What do we think? So no, they're not always made out of nucleic acids either. All right. So just a kind of reminder, so the type of catalyst or enzyme that we learned about recently was actually a piece of RNA. Okay. So just kind of go back through. She did talk about this a little bit in her notes on transcription and translation. So just make sure you look those over if you don't at least get through the bulk of the material. All right. Okay, so number 43. So where does glycolysis take place? So we have the mitochondrial matrix. Is that where it takes place? So what takes place in the mitochondrial matrix? Does anyone happen to remember? So that one is our TCA, or the Krebs cycle. All right. How about the cristae? Is that where glycolysis takes place? Sorry. So the cristae is also not where it takes place. This is where the electron transport chain occurs. All right. Our cristae is also our inner mitochondrial membrane. All right. Our outer mitochondrial membrane doesn't really do anything for us, unfortunately. All right. All right. And then how about the extracellular space? Is that where glycolysis takes place? No. How about the cytosol? Good. So our cytosol is where glycolysis takes place. All right. What is another way to refer to the cytosol? Good. So this is also our cytoplasm. All right. So she has either option. All right. Either one would be correct. All right. In which part of cellular respiration is the most energy made and by what process? All right. So first off, which part of cellular respiration makes the most energy? So our ETC, or the electron transport chain, all right, that's going to be the bulk of where the energy is made from. And what process does the ETC use? Good. So this will be our oxidative phosphorylation, and this oxidative phosphorylation is also going to be referred to as, so substrate level and oxidative are two different things, all right? So in the case, our substrate level phosphorylation is what occurs for glycolysis in the TCA. In the case of our electron transport chain, we have our oxidative phosphorylation, which is also referred to as chemiosmosis, all right? And this is referring to the flow of our hydrogen ion gradients, all right? So it's referring to the osmosis for our chemical. In the case, this would be our hydrogen ion, all right? So our chemiosmosis is the same thing as our oxidative phosphorylation, okay? All right. So don't freak out too much about your cellular respiration. It's not going to get too much deeper than that, all right? She is not going to try and get you guys too much on like products or any of that stuff, all right? Just make sure you at least know your basics. All right. So 45, what is the purpose of kinase? So we have methylation, catalyst for cell division, phosphorylation, signal to arrest cell division, or synthesis of proteins. Good, so it'll be our phosphorylation. In the case of for kinase, what is kinase? Good, so kinase is an enzyme, all right? So just kind of keep that in mind. So in the case for enzymes, they catalyze reactions. When we say phosphorylation, this phosphorylation is allowing us kind of to activate a reaction, all right? So in the case, if it's phosphorylation, we're going to try and activate the reaction, not inhibit it, all right? All right, bless you. So which of the following statements is or are false? So both mitosis and meiosis begin with a replicated diploid cell. Is that true? All right, so, and just kind of a reminder, all right, so for our meiosis, or for mitosis first, all right, we're working with our 2N cell at the beginning, all right? So think mitosis and meiosis, this is going to be what occurs after interphase, okay? So this is the cell we have after interphase, and in the case, these chromosomes are going to be replicated, all right? And we're going to form two 2N cells, all right? In this case, the difference is that after mitosis, they will be unreplicated, all right? However, in the case for meiosis, all right, we have our 2N cell after interphase, all right? These guys will be replicated. All right, here we're going to go through our meiosis one, and this will give us 2 cells, all right? In the case, these cells will only be 1N, all right, but they will still be replicated. Then we have to go through our meiosis 2. right? After that, we're still only going to have ends, right? However, now all of these cells should also be unreplicated. All right? So, after that kind of brief overview, so which of the following statements are false? So both mitosis and meiosis begin with replicated diploid cells. Is that true? So this is true. Does meiosis 1 end with replicated haploid cells? What do we think? Yes. So that one is also true. Both mitosis and complete meiosis end with unreplicated diploid cells. Is that true? This is not true. This is false. All right. So for this one, the difference is we do have unreplicated. However, mitosis ends with the diploid cell, where meiosis ends with our haploid cell. All right. So diploids. These guys are haploid. All right. And then does meiosis 2 ends with identical unreplicated haploid cells? Is this true? Are we sure? So just be very careful with the wording. So we have identical here, which would make this false. All right. In the case mitosis would be identical. However, for meiosis, these would not be identical. All right. Remember, we have a few things going on with meiosis that allow for genetic variation, all right, which causes these cells to not be identical. All right. Do we have to remember what causes them to not be identical? So for not identical, we have non-disjunction. All right. We also have crossing over. All right. So these are some of the reasons we would say that they're not identical. All right. So for mitosis and meiosis, we also have a few other things to think about, the types of cells that they begin with. So do both mitosis and meiosis involve gametes? So this would be no, they do not. All right. Does mitosis involve gametes, or does it involve somatic cells? Good. So somatic cells, these are also our body cells. All right. How about, so we also know that they both involve somatic cells. Meiosis doesn't involve somatic cells or gametes. So these involve gametes, or in the case, these can be referred to as our sex cells. All right. So kind of just the basis overview for the division process. When do chromosomes align along the equatorial plates? Good. So metaphase, and also this could be our metaphase plate. All right. So in the case, we also have when are chromosomes replicated. All right. So we have the S phase. All right. So our function of the S phase is replication, or also, we also learned about this aspect more recently. This would be our DNA synthesis. When else would we have chromosomes be replicated? So would chromosomes still be replicated in G2? Thank you. Would it still be replicated in the M phase? So what is the M phase? So our M phase, this would be, so in the case of our mitosis, this would be our mitotic or our meiotic for meiosis. All right, this would be our M phases. So would our chromosomes still be replicated in our M phases? What do we think? Okay, so they would still be replicated in M phase, all right? Make sure you kind of just review when we see replication happening, when do we see them to be unreplicated, all right? So just real quick, I'll kind of bring us back here. So if it's asking us a question about like numbers and replication, we're going to say that they're going to be unreplicated after they divide, all right? So unreplicated after they divide, so post-M phase. All right. All right. All right. So 50. When are chromosomes unreplicated? So before our S phase, we have G1. Is it always, are they unreplicated in G0? So good. So G0 would be a not always type of response, all right? If this was G0 directly out of G1, then they might be unreplicated. However, if this is a G0 at any other point, they would have to be replicated, so it's not always one or the other for the G0, okay? So in terms of structure, for centromeres, do they hold together homologous pairs? Good. So they don't hold together our homologous pairs. Reminder, our homologous pairs look like this. All right. Are they located on unreplicated chromosomes? Good. So they are located on unreplicated chromosomes. Do they hold sister replicated chromosomes together? What do we think? Good. So our sister replicated chromosome, all right, for our unreplicated chromosome, we would have this centerpiece being our centromeres, all right? Do centromeres pull sister chromatids apart during anaphase? No. What pulls apart our sister chromatids? So this would be our spindles. All right. We can also recall back a little earlier. We can say our microtubules, all right, which help us make these spindles. All right, so what is the result of non-disjunction? All right, so this would be one of the parts in our meiosis 1. All right, remember meiosis 1, we're going to see these guys being pulled apart. All right. So if they don't always get pulled apart, we're going to end up with non-disjunction, okay? So in the case of non-disjunction, are cells frozen at the metaphase plate? What do you think? No, they're not frozen at metaphase plate. We're going to continue with division, okay? Would a daughter cell be n plus 1? What do we think? So in the case, if these guys don't go ahead and divide, we're going to end up with something where one is going to have an extra chromosome, all right? So in the case of an extra chromosome, this would be what we consider N plus 1, all right? This the vocab word would also be aneuploidy in this case, all right? So we can have a daughter cell with N plus 1. Would we possibly have polyploidy occur? What do we think? So just a reminder, our polyploidy is 3 sets of chromosomes, all right? So one of the things, usually non-disjunction will not occur at all of the chromosomes. So in the case, we won't see polyploidy unless it's a very extreme case, okay? Polyploidy would usually be associated with something else. So would we have apoptosis occur with non-disjunction? What do we think? Do we remember what apoptosis is, guys? Good. So this is our cell death. All right. This would not be a signal for cell death. All right. So apoptosis will not occur. Would a triploid offspring be possible? What do we think? So, real quick. So aneuploidy, my bad. This would be N minus 1. Correction. Okay. Sorry. Our triploid would be our N plus 1. I wasn't reading my notes correctly. Sorry. So aneuploidy would be our N minus 1. N minus 1 would also be possible. Our triploid offspring would be our N plus 1, which is also the same thing as B. Okay? All right. So real quick, just kind of an example for triploid offspring. What examples did we see in class? So our triploid offspring example, we could see a child with trisomy 21, all right, which gives us Down syndrome. All right, so if she asks you a question for an example base, Down syndrome is usually the most common. All right. Which of the following correctly describes a karyotype? All right. So a karyotype assists in determining transmission of traits within a family. Yes or no? No. What does that describe? So this would be our pedigree. All right. Does a karyotype track gametes and predict offspring? Good. No. This would also not be true. This would be our Punnett square. Is it a visual profile of chromosomes in a somatic cell? Good. So keynote visual, our karyotype, all right, is the visual picture aspects that we can see of our chromosomes. Usually we'd see them from the somatic cell. You can also see them from a gamete, all right? They just look different in terms of number of chromosomes. And then offspring, potential for 2 different genes carried by a parent. What does that describe? So this one would be a dihybrid cross. All right. Okay. So in terms of combinations of gametes, so we have a genotype that's been given to us. All right. How can we determine how many combinations of gametes are possible? What should we use? Good. So we should use our 2n. All right. What is n? What is n? Good. So this is our number of heterozygous pairs. So if we go back through, our B, our D, and our H would all give us heterozygous pairs. All right. A would give us a homozygous dominant. F would give us a recessive homozygous person. And so in this case, we have 3. So 2 to the 3rd should give us 8. All right. So the answer would be D. All right. So the next couple give us a little more work to do. So she's guaranteed you a question about Munchkin cats. All right. So just remember that in the case if you have 2 large M's, this is considered lethal for Munchkin cats in general. All right. These will not be born. So in the case if we have 2 Munchkin cats, these Munchkin is still carried on the dominant trait. So all of these cats would have to be a heterozygous. All right. And so in the case for this cross, we should have 2 heterozygotes crossing. All right, so we should get a cross that looks something like this. In this case, this cat will not be born because, again, we said that is lethal. All right, and so in this case, we're looking for the probability that they will have a normal kitten. And so this kitten will be the only one born that will be normal. And this probability would be referred to as 1/3. All right. We also have another cross where if a woman has a child with a man who expresses blood type B and the child expresses A, which of the following is possible genotypes of the mom? All right. So in the case, before we really think about this too much, we know that dad expresses B. However, at this point, if the child's A, what does dad also have to express? What do we think? Thank you. So dad would also have to express O, all right? If he had 2 B's, in this case, the child would have to be a B, all right? So in the case, we know that the child is going to land upon here somewhere, and that the child has A, all right? So what does that mean mom has to give? Oh. Sorry. All right, so the child has A, all right? And so that means the child has to be along one of the dad's O's, okay? So in the case, what does that mean mom has to have? So that means mom at least has to have A, all right? And in the case, we can't really define what else mom has. However, in this case, from our choices, this means, oh, hold on. Okay, so from our choices, this means that mom could be AA, all right? She can't have B, she can't have BO. She at least has to give the child 1A, all right? In the case, mom cannot also have OO because she has to give the child at least an A. She could, however, be AB. She could also be AO. Okay? All right. So she's also guaranteed that you guys will have a calico cat question. All right? So in the case, calico cats, specifically, this would be sex links. Yeah. right? So in the case, a female calico cat should have the presence of XB and XO. If we go ahead and she mated with an orange male cat, that means the male has to have an O presence, and since he's male, he'll have to have a Y presence. All right, so if we go ahead and cross these, we get XO, XB, XO, XO. We have an XBY and an XOY. All right. So in the case, what is the probability of a female calico cat? All right. So in the case, we have to look at all of our females. All right. And so in the case, what is the probability of a female calico cat? That'd be 1/2, because only one of the two females possible can be calico. All right. All right. In the case, what would a genotype look like for a male calico cat? So kind of going through, so A and D are actually the same thing. So XB, XO would be a what type of cat? This would be a female calico. All right. How about an XB, XBY? This would be a black male. How about an XOY? Orange male. And how about XB, XOY? So this one would be our calico male. All right. So the reason you won't want to ever use a cross with a calico male is because they're rare, but they're also sterile. All right. All right. So we also talked about a particular gene. It's called SRY. All right. So which of the following is false concerning SRY? All right. So is SRY a sex-determining gene? Good. So that's true. Is it found on the Y chromosome? Okay. How about, is it found on the X chromosome? No. So this is false. And it can cause male features in a female if translocation occurs. So this is true. Remember our translocation, meaning we're moving a piece of the chromosome and putting it onto another chromosome. All right, so we're transferring it somewhere else. And so in the case, if it were to transfer onto the X chromosome, it's not normally found there, but it could cause them to have male characteristics. All right. So a little more concerning our structure. So for 60, anti-parallel refers to, and so we have the relationship between base pairs of the strands of a double helix. B, the relationship between nucleotides of one DNA strand. The relationship between side groups of the DNA backbones, or the relationship between the 5' end of one strand and the 3' end of the other. So it should be D. It's the relationship between the 5' end of one strand and the 3' end of the other. All right. That's how we get our anti-parallel. On the other hand, we also have complementary aspects. So complementary refers to which of the following? Is it the relationship between base pairs of the strand of the double helix? What do you think? So A is what complementary is referring to. All right, our base pairs, they're going to complement one another. So in the case, if we know we have A on the template strand, we know that the new strand should code for a T for a DNA strand. All right, that's the complementary aspect that we're referring to. Okay? So in that aspect, which of the which of the following is true concerning base pairing in DNA? So does A pair with U and is it connected by 2 hydrogen bonds? What do we think? Good. So this is not true. What structure would it have to be for A to pair with U? Good. So this would have to be for RNA. Does A pair with T and is it connected by 2 hydrogen bonds? Good. That one is true. Does C pair with A? How about does G pair with T? Does C pair with G? And is it connected by 3 hydrogen bonds? So that is also true. C and G will be connected by 3 hydrogen bonds. A and T will be connected by 2 hydrogen bonds. In this case, order does not matter. You could also say T is connected to A or G is connected with C. All right. All right. So, which of the following incorrectly pairs the enzyme in DNA synthesis with its function? All right. So, we have helicase unwinds the double helix. Is that true? Good. So that is true. That is the function of her helicase. Does DNA polymerase 1 remove the nucleotides from the 3' end? Are we sure? Okay. So DNA polymerase 1, this one is false. All right. It does not remove nucleotides. It removes the primer from the 3' end. All right. It removes primer, not nucleotides. So for the next ones, does topoisomerase stabilize and unknot DNA? Good. So that one is true. Does DNA polymerase 3, does it add nucleotides next to the primer? So this one's also true. So DNA polymerase 3 particularly works on elongating for DNA synthesis. So it's going to allow those nucleotides to keep being added. And then does ligase seal the DNA strands together? Good. So it seals the DNA strands together. It also seals the nucleotides next to one another together. It helps seal basically everything together. All right. So 64, which is the correct order of enzyme usage in DNA synthesis? So which one should come first? So first one should be our helicase. Should make the rest of the option pretty obvious. So B should be the correct answer. So we should see our sequence be helicase. Then primase will come in to add the primer. Our DNA polymerase 3 will come in to help elongate everything, and then ligase will end it. All right. So these options don't include all of the enzymes, but for the case, this order is the most correct. All right. One question for 64, does anyone know what SSB stands for? So if you see SSB, that refers to your single-stranded binding proteins. Okay? All right. 65. What is the primer made out of? So is it made from polysaccharides? Is it made from protein? Is it amino acids? Is it RNA polymerase? Or is it DNA fragment? Good. So our primer is going to be a DNA fragment. All right. And in the case more specifically, it's a short single-stranded fragment. All right, so it'll be a short sequence of the nucleotides for your DNA. All right, so we do expect to have a question that concerns your pairing and making sure you know how your percentages work. All right, so your A, T, C, G, all of that should add up to 100%. And so in the case, if DNA has a 32% of thymine bases, what percentage of guanine bases does it contain? All right. So if we have 32% of T, how much A should we have? Good, so there should be an equal percentage for A. And in that case, these two together should give us 64%, which leaves how much left? So that means we have 36% left, all right, for our C and our G. So if we go ahead and say that they're equal or divide by 2, that means our G and C should both have 18%. Okay. All right, are there any questions about that one? Just heard a little. Okay. All right, so for 67, RNA polymerase is involved in DNA synthesis, transcription, translation, or protein synthesis. What do we think? Good, so RNA polymerase is special for transcription, all right? In the case, it's very similar to kind of what we have for DNA synthesis with our primer, all right? But we don't use a primer. Instead, we're going to use the RNA polymerase, all right? Why are we using RNA polymerase? So in the case, if that was the kind of question, is because we're creating RNA, all right? So it has to be made for usage with RNA. All right, so 68 is kind of a long one. So this is kind of referring to you want to make sure you're using tRNA, mRNA, and amino acids in this kind of structure. So which is the proper sequence of events? So translation creates tRNA, which is then transcribed using mRNA with a ribosome subunit, resulting in amino acids, which creates proteins. What do we think? Good, so no, we're not starting with translation in particular, so that should kind of help set that up. So B, transcription creates mRNA, which is then translated using tRNA with a ribosome subunit, resulting in amino acids, which create proteins. How does that one sound? Good, so that one is our answer. So just kind of break down the last two. So we also will not be starting with amino acids. They're not going to be what gets transcribed, all right? And transcription does not create tRNA, all right? So you can kind of knock those ones out pretty early, all right? All right. So we also had some examples in usage. She could kind of go one of two ways for a problem. So I've given you examples for both types. So if you have an RNA strand with the following sequence, what would its DNA template be? All right. So it's asking you how you got this RNA strand from your DNA. Okay. So in the case, how should we start this? All right, so we have to remember that our, first off, so our strands should be anti-parallel, all right? So that means since this is written 5' to 3', we should have our 3' to 5'. All right. So in the case, we should also note that this is an RNA strand, but we're looking for a DNA template. All right. So your answer, so your DNA should have your thymine. All right. So when we kind of go back and pair these things, we want to make sure that instead of using our U, we're going to put in our T. So in this case, our G would go with C, our C would go with G, our U should become A, our A, instead of becoming U, should become T. All right? And finish it up, G, and then A. Oh, okay. All right, and so in this case, our answer should be C. All right. So on the other hand, we have, again, so this time we are starting with our DNA strands, all right? And from the following sequence, what would its corresponding mRNA be? All right. So mRNA, we're not really saying there's anything too special about here. We're still going to an RNA strand. All right, this is still going to be in the process of our 3' to 5' answer. All right, so in that case, that would also help us eliminate A and C. All right, and so for this case, if you just go ahead and do the first few, so we would get our T would become A, our A would become U, and C would become G. And so in that case, our answer should be D. All right. So 71, this, the number didn't get translated over, so this should have been from number 70. All right. So I'll push this up so hopefully we can all see. So I did kind of give you guys your amino acid template sequence on the last page. So in the case, we're looking to use this sequence. So for D, all right. So in the case for our first amino acid, the way we want to use this chart is, so our first letter should be on our left side. So we're going to start down here with our A. Our second letter was U. So our second letter is on top. So if we go down here and there are A, U, G. All right. So for our first one, we should have MET or methionine. All right. So we can automatically eliminate C and D. All right, and so the last question is, should we be in 3' to 5' or 5' to 3'? What do we think? So in this case, we're still going to use our same sequence aspect from our last one. So we should still be in 3' to 5' when we get our amino acids out. So our answer should be A. All right. So definitely make sure you know how to use your chart and make sure you know how to put your RNA to DNA or DNA to RNA. Okay. She could ask either kind of scenarios. All right. So for 72, your code refers to the what? So our code is going to be referring to our DNA. All right. In the case, our codon is found in the what? So our codons are going to be found in our mRNA. Remember, the codon is the 3 nucleotide sequence. All right, so there's no real order necessarily for the 3 nucleotides, so depending on where you start reading from, you may or may not code different amino acids. All right, so be very careful when you're looking at your structures, and make sure, you know, you're using the first